Hey everyone, this is the warm-up on the light-dependent reactions. I'm looking at the quad on page 222, and I'm going to explain the process that we modeled in class. But this way, those of you who are absent or those of you who just want a refresher course in this can go back and use this video. So the first thing that I need to do is establish what is happening here. We are looking at a process that is occurring in the chloroplast of a cell. And remember that our chloroplast is one of those organelles that we have evidence for being an endosymbiote. So it has the double membrane. It's similar to cyanobacteria, divide by binary fission. And just like in the mitochondria, where we had the two membranes, we're going to have two membranes in our chloroplast. And so in the mitochondria, when we were looking at the electron transport chain, we were looking at the inner membrane of that mitochondria. We're looking at the inside membrane of the chloroplast, but this time that membrane has a special name. We call it the thylakoid membrane. The thylakoid membrane itself is going to be a phospholipid bilayer just like we've had before, and we're looking at the difference in a concentration gradient and using that to create ATP synthesis, which is just one more reason why I get kind of upset when you guys call the mitochondria the powerhouse of the cell. And you're calling it the powerhouse of the cell because it's making ATP. Keep in mind that in glycolysis, we make ATP in the cytoplasm, and in the chloroplast, we're also going to be making ATP through a very similar process we saw in oxidative phosphorylation. So please just stop calling the mitochondria the powerhouse of the cell. In your mind, if you want to remember it that way, that's fine. But then on an essay or an explanation, you should be able to give me a lot more information than that. Okay, so our inner membrane of the chloroplast is called the thylakoid membrane. And so what we're going to see is something similar to the electron transport chain that we saw in the mitochondria, which means we're going to take electrons and we're going to move them down a biochemical pathway, and at the end there's going to be a final electron acceptor, just like we had in oxidative phosphorylation. But in this case, there are a few differences. I'm hoping that you guys are noticing the similarities between the mitochondria and the chloroplast, and that you're noticing the similarities between oxidative phosphorylation and these light-dependent reactions of photosynthesis, enough to kind of use that as your clue of maybe they share a common ancestor, or at least asking the question, do they share a common ancestor? So, in our light-dependent reaction, it's an electron transport chain. We're going to be moving electrons down this biochemical pathway. So the first thing that you have to ask yourself is, well, where is this electron coming from? Okay, so this thylakoid membrane has embedded in it these photosystems and a transmembrane protein, and that thylakoid is separating the compartments of the chloroplast. We're going to call this area up here the stroma, and this area down here the lumen. So this is the inside of the inside membrane. This up here is the space in between the outer membrane and that thylakoid membrane. So this outer membrane is the one that's believed to have come from the host cell. This is the original prokaryotic membrane itself. Okay, so with the stroma and the lumen identified, let's talk about the process itself. We know that this is going to be a electron transport chain. So the first thing that we've got to kind of make clear is, where is this electron going to come from? So we're going to take electrons from water. So we're going to split water. We're going to oxidize water. We're going to gain that electron, and we're going to make, in the process, we're going to make protons, and we're going to make oxygen. Eventually, this oxygen will join with another. Let's write that off to the side. And oxygen is diatomic, so this will join with another oxygen. And then this oxygen would be able to diffuse out of the membrane, out of the thylakoid membrane, out of the membrane that surrounds the stroma, and then either into the mitochondria, or even it could just exit out of the cell through specialized pores, and then aerobic organisms can breathe that in and use that as their final electron acceptor. But plants will use it to Plants will split the water, strip the electrons off of it. In that case, the water is getting oxidized, and so something else is being reduced. But in that process, that byproduct that we make is oxygen. So what's going to happen here is this is what we call photosystem 2. And photosystem 2 is a pigment. So the main pigment that you guys are going to be familiar with is chlorophyll. There's actually the different types of plants use different types of pigments. And so you'll have your main photosynthetic pigment, and then you'll have underneath that accessory pigments. And these pigments absorb light. That is their goal. And the more pigments you have, the more light you can absorb. So chlorophyll, you have chlorophyll A, chlorophyll B, you have accessory pigments, xanthophylls, carotene, you have the different pigments will give different colors, and at this time of year, you're able to see many of the accessory pigments. So chlorophyll being the main pigment, it reflects green and yellow light, absorbs red and blue. And so this is what you see when you look at plants. As plants begin to shut down because of the change in available free energy, you're able to see more of these accessory pigments, the yellows, the reds, the oranges, because chlorophyll is shutting down, there's no longer being produced. And so you're able to see those accessory pigments more, but plants have them. So in photosystem 2, this is think of it like a collection/reaction center, and it's going to absorb light. This is light provided by the sun, the ultimate source of free energy on Earth. And photosystem 2 is going to absorb best light at a particular wavelength at about 600 nanometers. And so sometimes you'll see this photosystem 2 referred to as P680, simply because that is the wavelength of light that it absorbs best. It's going to redirect light to what's called a reaction center, and that reaction center is where the electron will become excited. Now, if we were to take a look at this process, I'm going to try to cover all of this up. If we were to take a look at this process, electrons that are excited when they go to a higher energy state and then are allowed to fall back down will give off that energy in the form of light. So you see this with neon lights. So you're providing the electrons with electricity. The electricity excites the electrons, and as they drop back down to their ground state, what would happen is they would give off light. Well, plants aren't glowing in the dark. Some plants aren't. We're not counting bioluminescence trees at this point. But we don't see that. And the reason why is because as the electron becomes excited, so this is my excited electron, excited by the light that was redirected in this reaction center, that electron is captured and moved along an electron transport chain. So the electron is transported, and this is a transmembrane protein. So we're going to carry the electron to the next protein embedded in that thylakoid membrane. Again, don't worry about the name of this protein. Just know that it's a transmembrane protein, and it's a proton pump. And if it's a proton pump, what that's going to do is create a concentration gradient difference. Now, the one thing I don't like about this particular photo is they're showing here that ADP plus your inorganic phosphate is being phosphorylated to ATP. The problem I have with this picture is it makes it seem like this is all going on at the same time. This electron is being transported to a transmembrane protein that will pump a hydrogen. See if I can draw this somewhere else. We're going to pump a hydrogen into the lumen of your chloroplast. We're going to pump a hydrogen into the lumen of a chloroplast. We are creating a hydrogen ion concentration gradient, just like we did in oxidative phosphorylation. So once we have that hydrogen ion concentration gradient established, we can use ATP synthase, the very same ATP synthase that we used in oxidative phosphorylation to produce ATP as the hydrogen is moving down its concentration gradient from the lumen into the stroma. And we call that photophosphorylation, because we're going to phosphorylate ADP with inorganic phosphate. But the reason that we're creating, or the whole reason we can do that, is because we're utilizing the kinetic energy of the hydrogens created by the concentration gradient difference on either side of this membrane as that electron, which was excited by light, passes through a transmembrane proton pump. Okay, so we're making ATP through this process. The electron is going to reach the second photosystem, which is called photosystem 1. And this one is going to absorb light best at 700 nanometers. So we're going to call this P700. The electron is going to get excited again. This time, instead of it being passed to a transmembrane protein, it's going to be passed to a particular enzyme called NADP plus reductase. So NADP plus reductase is the enzyme that will take that electron, and we're going to use this enzyme to attach the electron to the final electron acceptor in this particular pathway, which is NADP plus. So this is going to get reduced down to NADPH. So in summary, we've created oxygen from the splitting of water to harvest an electron. We have two photosystems in place that are responsible for exciting that electron. As the electron is being moved from one photosystem to another, that electron is going to allow the creation of a hydrogen ion concentration gradient. That hydrogen ion concentration gradient will power the ATP synthase embedded in this thylakoid membrane, converting kinetic energy into stored chemical potential energy in that ATP. The electron ultimately is donated to the final electron acceptor, which is NADP+. And the enzyme NADP+ reductase is the one that's going to take that electron, take a hydrogen as well, and we're going to reduce NADP+ down to NADPH. The whole point of this process is to generate some ATP and to generate NADPH, which we're going to need for the second biochemical pathway of photosynthesis.